Hey guys, this is Shelby with the Roger Public Library. So this week for Full Steam Ahead, I want to talk to you guys about microorganisms. So this is kind of a big group, but the one thing that they have in common is these are organisms that you can't see with your eyeballs, right? You have to use a microscope in order to see them. And many different microorganisms are prokaryotes, right? So they're simple single-celled organisms that don't have a cell nucleus. But that's not all of them. There's also eukaryotic microorganisms. That includes some single celled stuff like yeast, which are a type of fungi, as well as things like algae or certain algae and other protists. But there's also multicellular microorganisms, which includes things like water bears, right? So that are more complicated. And microorganisms, because there's so many different types, they have a bunch of different roles that they kind of fill in the environment. So some of them are autotrophs, right? So things like algae or cyanobacteria that perform photosynthesis, and they produce oxygen, and they use the sun's energy to sustain themselves. But then there's also some that are decomposers, like yeast, that actually break down organic carbon and turn it into nutrients that other organisms can use to grow. Right? And there's also there's some microorganisms that are harmful. We call these pathogens. There's also some that are like parasites, and these perform important roles in the environment as well, right? Because they do, right? Because they actually promote genetic diversity, right? So if you have like a big population of like crabs or apple trees, and they're all the same, if there's not a lot of genetic variants, then they'll all be really susceptible to the same germs. But if there's more diversity among them, then some will survive and they'll be able to, and they won't all be wiped out by the same microorganisms. But an important point to make is that just because we can't see individual microorganisms with our eyeballs, is that sometimes they'll aggregate and form groups together. So like if you've ever been to like a creek or a pond and you see like slimy stuff growing, that's right. Those are microorganisms that are kind of stuck together into things called biofilms. And biofilm, right? It's just slimy stuff. The reason that these form is that a lot of microorganisms have like a sticky coating, and this causes them to get stuck to stuff like rocks and other surfaces. And then once they're there, other microorganisms will stick on them and they form like big mats and stuff. And sometimes biofilms can be problematic because you have all these different microorganisms, not necessarily the same species. You can have a bunch of different species all mixed together in these colonies, or sometimes people call them like microorganic cities, because so much is going on there. But they'll transfer genetic information between each other. Different organisms, different species will, in the form of little plasmids, right? So little donut-shaped containers with DNA in them. And this is called horizontal genes transfer. And when this happens, they'll pass on useful traits. But unfortunately, one useful trait for bacteria living in a mat like this is antibiotic resistance. So that means that we can't treat. So if you have like a bacterial infection and it forms a biofilm inside your body, then it can't be treated, or it's harder to treat it using medicine. But there's also circumstances where biofilms are useful and where we actually like cultivate them. So like there's wastewater treatment processes, like treating sewage using biofilms to filter out a lot of the harmful stuff in them before the water goes back into the environment. There's also certain things that are like but not quite biofilms that are formed by eukaryotic cells, right? So things that aren't bacteria, that are more complex cells. And a really cool one I want to talk to you guys is slime molds, right? So slime molds they are not quite actual molds. They're not fungi. They're kind of a weird hodgepodge group of organisms that aren't super closely related to each other. But they're all amoeboids. And so amoebas, they're single-celled organisms that don't have appendages. They don't have arms and legs. But the way that they move is kind of like by wiggling, almost like a slug does, right? So you think of them about like microscopic slugs. And slime molds are single-celled, right? But under certain circumstances, they can group together and form big, gross blobs called grexes, right? And so this is a slime mold that someone took a picture of in Tasmania, specifically Fuguloseptica, or dog vomit slime molds. And this, you can find this guy here in Arkansas as well. Not so much this time of year, but it's pretty common on like bark and dead, decaying organic matter in the summer and the fall in particular. Right, and so these grexes are they're weird because even though you have individual single-celled organisms, they're capable of communicating with each other using pheromones, right? So like chemicals that they release and that other slime molds will sense and then be able to act on. Right, so they can actually coordinate their movement, and because of this they can solve problems, right? So if you put a slime mold on one side of a maze and put a source of food in the middle, it'll actually find a path through the maze, and it can make like networks that if you have a bunch of different food sources laid out on a plate, it'll find the optimal path between them. So there's cool like things you can do with slime molds to make them solve problems, even though they don't have brains, right? It's just a bunch of single cells that are acting together. And if you want to learn more about slime molds, we have this really cool book. All right, it's Creeping Slime, or Slime Molds, by Ellen Lawrence. It's a picture of a slime mold growing, kind of making these really cool networks on it on the cover. But it has a bunch of really good pictures, and it also talks about some of the experiments that I mentioned, including one where slime molds kind of grew and branched out and created a network that resembles the Tokyo subway, right? Which is cool to think about, right? But you can actually make your own slime mold at home using something called floam. So floam is just slime that has foam in it. And this is one that I made a little while ago, maybe a couple weeks back. It keeps pretty well as long as you store it in an airtight container so it doesn't dry out. And the foam that I used for this was I crumpled up a styrofoam cup, which you can do that, but it will be a little bit lumpy. You can also use microbead filler, and that's what we're going to be using today. So first step is you get a bowl, and you'll want to measure out glue. I'm going to be measuring out half a cup because I think it makes a good amount of slime. I'm going to just pour this into the cup because I think it's going to take a long time if I wait for the pump. And of course, this is a little messy because it's glue, right? But it also cleans up pretty well. Just regular craft glue that you can wipe down with soap and water. So that's roughly, making a very big mess. That's roughly half a cup of glue. I'm going to put that underneath there. This is a lot messier than I thought it would be. And now we're going to add in some microbeads, right, so that we can get kind of a bumpy texture, like we saw in this picture over here. We don't need a lot. And these are also pretty messy. That's mainly because there's a lot of static electricity built up in them. If you do use like a plastic cup that you or a styrofoam cup that you crumble up, it won't be nearly as messy as the microbeads are. And we're going to try to get a nice color, a yellow color. I'm going to add a drop of green and 2 drops of yellow, right? Because when we look at the reference picture that we have, it's kind of an electric green, right? Or an electric yellow, kind of somewhere between the two. We're mixing them together, right? And so we have this really nice, it almost looks like radioactive, really. Okay. And then we're going to add some liquid starch. I'm going to start off with a tablespoon, but I think we'll need a little bit more than that. All right, so it's starting to get the nice slime texture that we know and love. And one more tablespoon. And I've made a lot of slime over the years, and it's not really something that you need to measure. You can kind of eyeball the ingredients. And the exact consistency doesn't really matter as much as just it not being sticky anymore. So if you don't have measuring cups or you don't want to use them, you shouldn't feel bad about it. Right, so the actual chemical thing happening here is that the glue is made out of polymers, right? So, it's made out of little organic molecules, right, called PVA. And when we add the liquid starch, it creates new bonds between the individual molecules in the glue, right? It cross-links them. And that gives it kind of stretchy consistency. And it's what makes it not sticky anymore. Eventually, once you add enough, which I don't think I have. Right. And so with this experiment, the slime itself is easy to clean out. The thing that might be difficult and that might stain surfaces and clothes is the food dye. Yeah, there we go. This feels pretty good now. I have no idea how much liquid starch I ended up adding to the half a cup of glue, but we did get a good consistency. And you can see the nice little bumps, which kind of resemble the little bumpy, spory things that are in slime mold cracks. Right? Yeah, so that's fun, more or less. Now for our now onto our experiment. The way that scientists will study microorganisms is normally through something called a Petri dish, right? So this is a little flat glass container that is at really convenient size, and it's also easy to slide under a microscope. And these will be filled with something called agar, right? And agar is made out of red algae, and it's kind of gelatin-ish, right? It's kind of a jelly substance. But the difference between gelatin, which is what we have here, and agar is that agar is made out of a sugar called agarose or agarose, and gelatin is made out of a protein called collagen, which is extracted from bones and tissue of livestock, like cows and pigs, for the most part. So, right. And then agar is usually mixed together with like nutrients in the form of like say broth, just to help the microorganisms grow, right? So they have a food source. So right there's not much to this. I kind of just made the gelatin according to the package and put it into these containers, which have nice lids, meaning it's easy to store them. And theoretically, you could use flavored gelatin like this to grow microorganisms. But the problem with that is that this has different preservatives in it, and that might affect how many microorganisms you're able to grow or what kinds. So if you wanted to do this, you would probably want to use unflavored gelatin and mix it in with broth of some sort. This one I added some sprinkles to because I thought they had a nice shape. A lot of bacteria are like bacillus shape, right? So they have this nice rod shape, similar to sprinkles. But it didn't work out super well. The dyes in the sprinkles kind of bled out into the jello, and so it looked a little funky. right? But the reason that agar is preferred over gelatin in a lot of situations is that agar is a higher temperature point, right? So if you've had Jello before, you know that when you put it in your mouth, it kind of melts right away. But where right, so its melting temperature is lower than our body temperature. Whereas agar, it doesn't melt until about 185 degrees Fahrenheit, and that means that a lot of bacteria and my other microorganisms that prefer an elevated temperature are easier to cultivate in agar because it won't liquefy the same way that the gelatin does. But if you're growing things at room temperature, that doesn't really matter too much, right? And of course, just point out, we're not growing any microorganisms today, but next week we're going to be doing an experiment that does involve actually growing germs. So you probably want to check that out. And of course, there is an exception to the types of things that you can grow in agar plates, and that is viruses, right? So viruses kind of break the rules, where organisms for the most part are cellular, and viruses aren't. They don't have a cell structure at all, and for that reason, some scientists don't say that viruses are organisms. Instead, they're kind of just strands of DNA encapsulated. They have like a shell around them, and they aren't capable of replicating on their own. So they have to have a host cell in order to replicate and make new copies of the virus. So one thing that you could do if you were a scientist studying a virus, is you could grow bacteria in a plate, and then put the virus in. And certain viruses called bacteriophages will use the bacteria as their host cells, and they'll actually grow in that medium. But in other cases, you have to use things like chicken embryos, so fertilized chicken eggs, which it's kind of gross to think about, but it is really important, right? This is like a key step in manufacturing the influenza vaccine, is growing the influenza virus in chicken embryos. So even though it's not fun to think about, it is a really important step that is used to keep a lot of people healthy from getting infected by the flu every year. But that's what I have for you guys this week. I hope you guys had fun, and I'll see you next Thursday.